Hello everyone. We're going to start soon. Before we begin, there's going to be a link in the chat to a document. It's going to be a Google Doc where you can ask questions, and it's anonymous, and I'll just be looking at them from time to time, and you can ask them as we go along. I'll try to answer them to the best of my ability. Okay, so this week's topic is going to be all about muscles and tendons. And yeah, so it's going to be the structure, the healing, and their histology. Okay, let's move on. So we're going to start with contents. So we're going to talk about the muscles first. We're going to talk about the macroscopic structure, microscopic structure, and anatomy of the sarcomeres. We're going to move on to then to talk about the tendon, composition of the tendon, structure of tendon, and the cells of the tendon, and tendon healing. And we're going to do some sample questions and answers, and then a Q&A session. So let's start with muscles first. So muscles, just like tendons, as you're going to see, they're formed in a very similar way. And the key thing here is to remember that it's packets, right? So you have the skeletal muscle itself, and you have these muscle fascicles. So a fascicle is one of these components. And each of these components are covered by different things. So we have the epimysium, which covers around the muscle itself, so it covers the muscle. Then we have the perimysium, which is wrapped around the fascicles themselves, and mysium and epimysium and perimysium, they're all connective tissues which help support the tissue. And then we have fascicles, which is the bundles of the muscle fibers, right? And they're, as you can see, they have these cells. And they allow, and the reason you have these fascicles, and so many of them, is because they allow for specific activation of muscles, which gives you the power and the accuracy. So for example, if in your back muscles, which are one of the more powerful ones, if you want to pull something, but you don't want to pull it too hard, like pulling a door, then use certain activation. However, if you're trying to pull yourself up, you'll recruit much more muscles. And the reason this is because you have these muscle fascicles. Now, if we dig a little bit deeper, we'll see that there are also there's also an endomysium, which is the muscle fiber itself. Now, the endomysium is the connective tissue. It's a layer of collagen, and it's inside each follicle. Now, the muscle fibers themselves, they're long and multinucleated cells, and they're made up of myofibrils. And these fibrils contain the sarcomere units. Now, the myofibrils themselves also have mitochondria attached all over them to provide energy. They have the sarcolemma, which are supportive tissues, and sarcoplasmic reticula, which provide calcium storage, as you can see right there. Now let's zoom in a little bit better for the skeletal muscles. What we're looking at here is different types of muscle. We have smooth muscle, we have cardiac muscle, and we have skeletal muscle. Now the key structure here is that you have these striations. You see how striated this muscle is? Super striated. Whereas the cardiac muscle has some striations, as you can see here, but they're not that prominent. And then you have smooth muscle, which is a bunch of cells. They have no striations, and they just sort of are in a random place. Now, with the skeletal muscle, as you can see, there's many nuclei for one cell. So all this here is one cell, and there are many nuclei. Now, the smooth muscle has a lot of cells, which are mononucleated, and cardiac muscle has cells which have one or two nuclei. But back to the skeletal muscles. So the striations are the myofibrils which are running parallel to the cell. Sarcomeres themselves are functional units of each myofibril. The shortening of these creates tension, and the tension eventually over the whole muscle creates a contraction. Now we have different organelles. We have mitochondria, which are used for energy. We have a sarcoplasmic reticula, which is for calcium storage. Terminal cistern, which require the calcium from the sarcoplasmic reticula to release calcium. and T-tubules, which conduct action potential of the muscle. Now let's look at the sarcomere itself. So a sarcomere is a functional unit of muscle. I'm sure everyone has seen these before, but let's break it down a little bit. The thick filament, which comprises the A band, is made up of myosin molecules which bind to the actin. And then we have the thin filament, which is made of actin, and it's a tropomyosin-troponin complex. And basically what this means is that this complex is wrapped around the actin. And once the tissues become ready for contraction, this is in the presence of calcium and of ATP, the actin becomes exposed and myosin heads can make attachments. Now moving on, we have different parts of the sarcomere. So the A band is the size of the myosin heads, which does not change during the contraction. The way you can think about this is that these muscles move inwards, whereas this stays the same. So you would see this gap here becomes closed, so this moves in that direction. So they stay the same, but these move and they slide onto each other. That's called the sliding filament hypothesis or model. Now we'll move on to the I band. So I band is the distance from one end of the thick filament to another. And as you can imagine, these shrink as the contractions become more prominent. And during a full contraction, this I band becomes non-existent. We have the H zone as well. So the H zone is the distance between two thin filaments. It becomes smaller and smaller and non-existent to the point that it sort of overlaps during a full contraction. The Z line or the Z discs are the borders of sarcomere. Now, if you look at this from on a microscope or from a histology sample, you'd see many Z lines, and all of these would demarcate one sarcomere, another sarcomere, and they would be all next to each other. So that's the anatomy of the sarcomere. That's all for the muscles. Now we're going to move on to the tendons. So we're going to start with the composition of a tendon. So the tendon is roughly 80% extracellular matrix and 20% cells. Now the tendon is very different from the muscle in the sense that you don't have, it's a structural tissue, it's a connective tissue, whereas muscle is a functional tissue. So the tendon is a connective tissue which helps facilitate the muscle. So if we look at it, it's extracellular matrix, which is 80%, and 70% of this is water, right? And 30% is solids, mostly being collagen type 1, ground substance type 3, and elastin. Now the point of this is so that it attracts water. So the solids, they're very negatively charged, right? Collagen type 1 forms these massive complexes which are negatively charged. Ground substance, hyaline, all this other stuff makes negative charges, and that attracts water. Now when you attract water, it makes it so that the tissue becomes much more stronger, much more tensile, and it's also much more flexible, which is what you need in the tendon. And this is achieved primarily through collagen type 1 and elastin. The rest is cells. So we have tenocytes. So tenocytes are, if we remember back to the osteoblasts and osteocytes, which are basically cells that create things, and they create the tissue, and they're found in the bones. Teno means tendon, so these are cells found in the tendons. We also have stromal cells, and stromal cells are the supportive cells of the tissue or keep the tissue itself. And these cells are super important in regeneration and secreting factors and in maintaining the tendon the way it's supposed to be. Now, as I said before, collagen and ground substance, negative charge molecules, the purpose is to attract of the ECM is to attract water. Now, collagen type 1 tendons, because they have collagen type 1, just like in bones, but since it's applied differently because of the water there, and it's packed differently, it has the highest tensile strength of all connective tissues. And the reason this is because they're packed together. So tendons have nearly the same tensile strength as steel, which is pretty amazing. And the reason this is because they're closely packed together and they're all arranged in the direction of a force. So if you have something closely packed together, right, with all these fibrils in the direction of the force, you have law of strength. So let's look at the tendons themselves. So macroscopic structure of a tendon, we've all seen pictures like this. So the tendon itself has three main parts. So you have the middle part, which is the tendon itself. You have where it inserts into the muscle. So that's called the musculo-tendon junction, or the MTG. Or where you have the bone insertion, right? So what do tendons do? They connect muscle to bone, right? And they facilitate the movement of the muscle. Without the tendons, your muscle would contract, but it wouldn't be able to pull anything. And the bone insertion is called the osseo-tendinous junction. Now, the way it inserts into the tendon, so if you remember from our previous session on the bones, we said that there's a periosteum, which is like a wrapping on the bone. Well, the periosteum serves an important purpose because it connects the bone to the tendon, which connects to the muscle. And this is where the Sharpie fibers come into play, which we also mentioned before. They provide nutrients, but they also provide a bone insertion. And the way this works is that you have the tendon, you have your epimysium, as we mentioned before, and the endomysium and the perimysium. But within the tendon itself, you have the peritenon and the other layers which help increase the strength of it. So here we see that the peritenon and the wrappings of the periosteum are continuous with the myofascia. And what this enables is that it's elastic, it's strong, and it covers the whole thing. Now, if you think of a tendon rupture, usually you get a break somewhere here, or it gets from the bone. So if it's like really traumatic, right? If something ripped off, sometimes you can pull off the bone because it's so strong, or sometimes you can just have it snap here and it just pulls off. In many cases, for example, if you have a biceps tendon tear, which can either happen here or here, and it snaps, it's like a loud snap. The tendon sort of becomes shriveled up, and the bicep would sort of just pull back up. The reason it does this is because the muscle itself has tension, and the tendons have tension, and they ensure that the muscle is kept strong and functional. The tendons themselves also are quite important because they contain, or they help facilitate coordination of proprioception within the joints. So let's look at the hierarchy of tendon tissue. It's very similar to what we saw in the muscle, right? So let's talk about the interfascicular matrices. So these are quite important. So you have the fascicles themselves. So this here is the fascicle, right? But between the fascicles, we have these matrices. Matrices are just a fancy way of saying tendon tissue. So these matrices, what they essentially do is that they help with the gliding, right? So just like in the epimysium, in the muscle, which exists so that it doesn't move around different organs, right? So let's say you have your muscles, and then you have skin, and you might have fat, you might have bursa. That gets in the way, right? And you have bone there as well. That all gets in the way. So that it moves smoothly, you have all these coverings, all this connective tissue. Now, each fascicle, in order to function, has to have its own little connective tissue. And this interfascicular matrix provides elasticity. So let's start with the paratenon, which covers the whole tendon. It's elastic tissue outside of the epitenon, which we'll get to later, and it enables movement of tendon against neighboring tissue. So like I said, this could be a bursa. So if you imagine, if we go back here, you might have a little bursa here, not here necessarily in this joint, but you could have like a bursa there. Or let's say you have a tendon here in the forearm, you might have a bursa there. And the thing is, you don't want the tendon irritating anything, right? And in order to have it nice and thick and be able to move, you have these connective tissues, which are such as the paratenon. The endotenon is what's wrapped around it's a connective tissue sheath, and it's what is wrapped around the bundles. Now, what this ensures is that you have gliding of the bundles against each other. So the more it gets stretched out, right, the more of these individual wires or these individual fascicles can stretch and glide. If there was friction, this would make it much more difficult to pull muscles when you contract, to pull the bones, but it also would make it much more difficult to resist tension. So what these connective tissues do, they enable the gliding of these bundles. Then we have the epitenon, which is continuous with the endotenon, and it sheaths the whole tendon unit. So whereas the endotenon could just be on one thing, the epitenon sheaths the whole unit. Now let's talk about fascicles. So fascicles is what the tendon structure is made of. It's very similar to what we saw with the muscle. So we have tendon fascicles, which are a group of collagen sort of fibers. So it's these things here. And there's different types. So you can have a primary fiber bundle, which is called a subfibril, a secondary fiber bundle, which is called fibril, and a third bundle, which is the whole fascicle. Now, you don't really need to know this. This is just out of interest, and it sort of to help you organize your sort of thoughts about tendons and muscles. It's just the different groupings. And I'm going to show another picture here. So this is another way to look at it. So you have your fibril, which composes a fiber, which composes a fascicle, right? And that composes the tendon. So the collagen fibers themselves, there are groups of collagen fibrils. So you might be asking, what's a collagen fibril? So a collagen fibril is the smallest possible unit of collagen, right, which contains tropocollagen. And this tropocollagen is just the collagen itself aligned together in the right direction. So we all know collagen, super strong, and you just pack it in these straight lines. And it's parallelly packed, gives it a lot of strength, and because of all this strength, the collagen gives it the tendon so much tensile strength and flexibility. And this is the reason for this is because it's parallelly packed. So if you want to think about it, it's like steel beams, right? If you have a concrete and you put steel beams inside and you put them parallel, you make the structure very, very strong. So we talked about the extracellular matrix. We talked about the way the tendon is, the hierarchies of the tendon. Now we look at the cells which produce this. So first thing here are some tenocytes that I cultured. And as you can see here, these tenocytes have yet to attach to their matrix, right? They're sort of free-flowing. This is what we're looking at right here. They're free-flowing. Some have little sharp blades like this, but they're mostly quite circular. You know, they haven't attached yet. Now here are some tenocytes which have matured. As you can see, these beautiful blades, nice and long, and they attach. You know, much like when you have the osteoblasts, which were circular, and the osteocyte, which had these many projections. Tenocytes are very much similar. And the reason they're so similar is because they're basically fibroblasts, which is a type of cell which produces ECM. And if it's in the bone, it's called an osteoblast. If it's in the tendon, it's called a tenocyte and tenoblast. So tenocytes and tenoblasts, these are specific fibrocytes and fibroblasts found within the tendons. A tenocyte is a mature tendon cell that is anchored to the collagen fibers and found throughout the tendon structure, whereas a tenoblast is an immature tendon cell that is not attached to any collagen fibers. And this is a sort of schematic to help you understand. So this is the stem cell, you know, this is when it's in its most primitive phase. You don't need to know all these factors here, this is not necessary to know. But what I'm trying to show here is that under certain factors, such as TGF-beta, they become tenoblasts, so you see. And then you have tenocytes is when it becomes attached to the collagen fibers and fibrils and helps maintain the structure. Much like the osteoblasts, which, you know, they're not attached, but once they become surrounded by tissue and osteo, they become osteocytes. It's the same thing. I'm just trying to show that there's a sort of consistency between the two. So we have a tenoblast, which becomes a tenocyte, and tenocyte is what maintains the whole tendon. It gives it structure and integrity to the fibrils. Now, we move on to healing of tendons. So a normal tendon has these beautiful parallel lines. You see how this is so well organized? Nice and parallel, right? That's what a normal tendon should be like. But a tendon that's healing is very much disorganized. You know, you have tissues all over the place. You have neovascularization, which is new blood vessels forming, and you have inflammation. Like when you can see, this is nice and clear, and this is very much yucky. You don't need to be an expert in histology to tell the difference between these two, right? This is nice and normal, but this is sort of disorganized. Well, there's a reason for this. So for the first 7 days of a tendon that's damaged, you have inflammation, right? So this inflammation from the damaged tissue, this is the tenocytes themselves just bursting out and releasing gunk, basically. Inflammatory substance, and this is your cytokines, this is your chemokines, and this is your TGF-beta, all these things. Now, that's the first 7 days. And around day 3, tenocytes start producing collagen type 3. So if we remember back, it was type 1, which is the normal tendon, produces type 3. Type 3 is much, much weaker, right? Type 3 is the cheap version of type 1. That's the way to think about it. It's there because it's quick to build, it's easier to build, and it's not that specific. So many other cells other than tenocytes can produce these cells. So you have collagen type 3. Eventually, this collagen gets resorbed, it makes scaffolding, and collagen type 1 can be added, right? So similar to how the bones heal, you put the fibrocartilage first, and then the osteoblasts come in and they secrete osteo. This is very much the same thing. Collagen type 3 is the weaker structure, which provides a scaffolding, and then type 1 needs to come in to sort of finalize everything. Now, the collagen fibril, this is called the remodeling, is at day 28 to 100 days, and the difference between the collagen type 1 just being deposited and the final product is that the collagen fibrils, they cross-link, right? And by cross-linking, this means they form bonds and they become stronger. And this is what gives the tendon its unbelievable tensile strength. However, once a tendon is healed, it's not the same as before, because either A, there are defects in the tendon, which have not been healed, B, because the tendon itself has very poor vasculature. So if you think about it, most of the vasculature goes into the muscles rather than the tendons. Tendons are quite avascular. And what you need for healing is you need the cells, you need nutrients, which is from blood supply, and you need blood supply so that they can take nutrients and take the waste out. Tendons don't have this. They're quite poor, they're quite avascular. And the reason we can tell the tendons are avascular is because they're quite white. You don't see any, muscles are quite nice and juicy, but tendons, they're quite white and they don't have any vasculature coming through. Now in tendinopathy, which is damage to tendons or tendon injuries, the healing process is stuck at collagen type 3, right? So that's a tendinopathy. So in tendinopathy, you have collagen type 3 instead of collagen type 1. And as we said, this collagen type 3, it's not meant to be functional. It's the weaker version, it's the cheaper version of collagen, which is more easy to produce, but it does not have the same strengths. And secondly, the collagen type 3 is not in nice parallel lines, as we can see here, right? As you can see here, it's not parallel, whereas here it's nice and parallel. So collagen type 3 is just deposited there so that the tenocytes with collagen type 1 can produce them later on. So we've covered that. I'm going to look for any questions. If anyone has any questions. Okay, just submit them through the Google Doc. If not, we're going to move on to the questions we have here. Some questions are prepared. This was a question in my exam in first year, which was, four years ago now. And it asked, what is shown in the following picture? So yeah, if people want to ask questions, just go ahead. And I'll see them, and I can answer them as we go on. So what is shown in the following picture? So this is from a microscope. It's a photo of a microscope. And what are we looking at? So it looks like a sarcomere, right? That would be like a good guess. It looks like a sarcomere. But we have to be careful. It's a specific way the sarcomere is formed. It's a contracted sarcomere. So if you just wrote sarcomere, if it was only sarcomere, you would probably get half a mark. Or 0 marks. So the important thing is, if you see one more question like this, which seems a bit too difficult for what it is, you have to be specific. So it's a contracted sarcomere. So how do we know it's a contracted sarcomere? Well, that's quite simple, because we can see the overlaps here, right? We see the vanishing of all the different lines. So the A band is still there, but there is no I band, there's no H zone. It's all missing, So all of these things are missing, and we don't see them there. We see the Z line here, right? We see the A band, but we don't see anything else. So this must mean that the sarcomere is contracted, right? So it went like this and it's contracted. So careful there. So now this is a sarcomere which has not been contracted. As you can see, we have the A band, we have the I band here, right? We have everything there. Then we have the Z disc. So as you can see, this is a non-contracted one. This is also a question in the previous exam. And it's asking us to, as per the sliding filament model, which one of these will change? So the sliding filament model is, so that's how we answer this. The sliding filament model is the model by which sarcomeres contract. And if we move on here, we see that A is the A band, which always stays the same. You know, it's a thick filament, right? So this is your A band, which always stays the same when it's uncontracted. B is our M line. So this here is the M line. And that sort of remains, as we saw here, the M line remains. That's M line. And M line demarcates the middle of the thick filament, right? It's the middle, and it's also the middle of the sarcomere. Moving onwards, we have C and D, which are the same. They're basically the Z line or Z disc. What happens to these is that they move closer. So this can be quite tricky because you could assume that this here could also be the I bands or the thick filaments. So you have to be careful here. It's always going to be the thick filament, which is really concentrated in this area. And the thin filament, as you can see, is barely visible. That's why they call it a thin filament. And once they slide together, they all become very thick. So if you see white lines like this, white areas, you know that's an uncontracted sarcomere. But if they're put together and contracted and there's no white lines, that means it's a contracted sarcomere. Moving onwards, E is the I band, which gets smaller because the thin filaments are brought closer together. So think about like interlocking fingers. And F is the H zone. So the H zone becomes smaller and smaller until it becomes non-existent during the contraction. As we saw here, you see the H zone is not visible. And this is just to demarcate this. Let's see if I can hide these things. Yeah. So as we can see, this is an uncontracted sarcomere, uncontracted muscle. Once you get the contraction, they overlap. You see the thick filaments, they overlap like this. And when they overlap, they become really close. The I bands are no longer there. The I space is no longer there. And the Z lines are still there. And we have the thick filaments. So I think this is quite key to understand the model. Even if you don't understand the physiology of it, which is anatomy, we don't really go into the physiology. But what's important is that you bind and you move them together, and that's what causes your contraction. That's what a contraction looks like. And of course, this has to be in the presence of calcium and ATP. Now I'm going to say, learn your sarcomeres. Sarcomeres always come up in first year exams, and it's a key topic, right? It's a key example of a year one topic. So if I were you, I would learn your sarcomeres. And it's not enough to know the physiology, you also need to know what it looks like under a microscope. And this is what caught out a lot of my peers, and even me, because I did not know what this looked like. I never saw this sort of picture. But it did come up, and it surprised everyone. So know what a sarcomere looks like under a microscope. This is the model, right? They might ask you to draw this, which is quite easy marks. You know, you just draw the whole thing out, and you label it. But this, I think, is very key to know. So it's a little bit different. It's the application of the knowledge. It's the application of the anatomy. Okay, more histology. So who dislikes histology? Everyone in first year, because it's never really well explained. So we have 3 different slides of skeletal muscles. I mean, they don't tell you that, but that's what this is. So sorry, 3 different types of muscle. And we have to label them and what their features are. So let's start with the first one. So the first one here is smooth muscle. How do we know that? No striations and 1 nucleus per cell. And you see they're not in a sort of strict pattern. They're sort of all over the place, and they're quite bound together, and you have a lot of tissues in between, and you have all these blood vessels and all this other stuff. Smooth muscle is found primarily in connective tissue, right? CT, connective tissue. Or in stuff like your gut, where you don't have striations. Sphincters, if it's the part that you don't control, so like inner sphincter rather than external ones. Stuff like the stomach, digestive system, where you have peristalsis, anywhere where you have involuntary movement, that's smooth muscle. And smooth muscle, no striations, right? And it's a single nuclei. The reason it doesn't have striations is because it doesn't need striations. Striations are to increase power, right? You don't have that. It's to be an automative mechanism, an automative movement. That's why you don't have striations. Second is cardiac muscle. Now, cardiac muscle, we have to be careful. It looks like a Y shape. I think that's what they teach you. It looks like a Y shape. And we can sort of see this here, like so. Right? That's one cell. And we can sort of make it out here as well, like this cell. And sort of here as well. This cell here. So these are Y-shaped, so it's cardiac muscle. You have slight striations, if you can see them here, the striations right here. But they have things called intercalated discs, and these are your intercalated discs. This is your intercalated disc. This is your intercalated disc. Yeah? So all these intercalated discs, they basically are specific to cardiac muscles. Another thing with cardiac muscle tissue is that you have 1 to 2 nuclei per cell, which is very different from striated skeletal muscle, which has it has a lot of striations and a lot of nuclei. And compared to smooth muscle tissue, it only has 1 nucleus, but no striations. Finally, it's the skeletal muscle, and it's pretty obvious here. The striations are right here. You know, it's well striated, and they're parallel. They're multinucleated because we have 1 nucleus here, 1 nucleus here. This is all just 1 cell. And yeah, so this is how you basically figure it out. If you see striations, think skeletal or cardiac, and then you look for the shape. Is it a Y shape? It's cardiac. Are there a lot of nuclei? Skeletal. Are there 2 or 1 or 2? Then it's cardiac. And if it's just 1 per cell, then that's smooth muscle. Do be careful though, if the cell is undergoing mitosis, it might appear that it has like a bigger nucleus or maybe 2 nuclei because it's splitting. But be careful, it's not 2 nuclei, it's just cell division, but you're unlikely to see that with muscle tissue. That's just in general for histology. Maybe we should do a video just on histology, like the basics of principles of histology. Okay, question for cells, muscles. Now we do some tendon questions. So it's describe the structure and function of a tendon. Okay, so that was 4 marks. This is a very typical question. So we answer it quite simply. We start with saying, what is a tendon? Right, so we do like a brief definition. So we do a definition. So we do a definition here. And we say a tendon is a dense, regularly arranged connective tissue, right? Because it's parallel lines, right? Parallel lines, think about tendons. And attaches bone to muscle, right? And facilitates movement. So that's the function of it. So we talk briefly what it looks like about the structure, and then we mention the function. Then for tendons, we talk about the composition, which is 80% ECM, 20% cells. And of that 80%, 70 is water, 70 is solids. And we talk what the solids are. We can also say how the structure is adapted to the function is because of the high collagen type 1. So high collagen type 1 means that we have a lot of tensile strength of the connective tissue. Then we also have that collagen fibrils are arranged in bundles in the direction of the force, which helps the function, which is to facilitate movement through contraction muscles. And we also say how they attach onto the bone. Now this may seem like quite a lot for 4 marks, and you're right, there's 1, 2, 3, 4, 5 points made. But it's always good to be thorough. They might ask just about the composition of tendons, or they could do like describe the different composition of tendons, cartilage, and bone, and you have to talk about that. But I think this is a sort of comprehensive answer, and whenever anyone asks you about tendons or the structure and function, this is what should be in your mind, right? You think tendon, okay, it's it's packed tissue, parallel lines, a lot of ECM, not that many cells, a lot of it is water, high collagen type 1, and collagen fibrils in the direction of the force. So that's what I will be sort of thinking in my head about the structure and function of a tendon. So moving on, we have tendon healing. Now, if you watch the previous video, or even this one, it's very similar to how a bone heals. So we're going to be using the inflammation, repair, and remodeling method of thinking about things. Yeah? So it's inflammation, repair, and remodeling. This is anything with musculoskeletal, right? Get inflammation, you get repair, and then it has to build up back to what it originally was. With tendon healing, it's a little bit more specific here, because just like in the bones, we had the whole macrophage co-stimulating factors, and we had rank L, we have specific molecules for tendons. Now, there's many molecules, right? There's many cells involved, especially in the inflammation. And we're not going to cover all that. We're just going to stick with the basics, right? So the first thing is that you have PDGF. So this is platelet-derived growth factor. So if you see GF, that's a growth factor. And then we have TGF-beta as well. So platelet-derived growth factor is for chemotaxis. What's chemotaxis? So basically, the cells need to get to a place, right? They need to migrate. How do they migrate? It's by tissues releasing this dangerous chemical or this attractive chemical called PDGF and TGF-beta. TGF-beta helps facilitate the differentiation of the tendon cells, as we saw before. Now, when we have inflammation, this is the first 7 days, we have cells from epitendinous tissues. So this is tissues like the periosteum, epitendon, endotenon, paratenon, right? All these cells from all these other tissues, they sense danger. And they migrate to the area of inflammation. Now, the defect, let's say you're exercising, right? And you have a sore tendon. Or let's say you take a medication and that affected your tendons, right? It's inflammation. There's some sort of problem to your tendons. And you get granulation tissue, which is basically a vascular type of tissue. And you get matrix proteins laid down as scaffolding for collagen synthesis. So your matrix proteins, this is what we talked about, your ground substance, your elastins, your collagens, that sort of stuff. And this is sort of, these matrix proteins exist prior to the collagen synthesis. They have to get the setting ready. So if you think about it, inflammation, think like a natural disaster, right? Someone needs to call the police, someone needs to call the fireman, someone needs to call the ambulance, right? This is what that is. And then the inflammation is the danger signals. Now the tenocytes are all set up. You need to repair the area, right? So this is them setting things up. So you have tenocytes, which are specific cells, and fibroblasts. They get to the zone of injury. Tenocytes and fibroblasts do what they do best. They secrete stuff, right? There are chemicals there are cells that create things. So this is your collagen type 3 initially. And the key important thing here is that it's collagen type 3 in a random orientation, right? Think about it like this. It's a disaster, there's problems, and they just try to do the first best thing they can do, right? It's the first aid, yeah? Is it going to help? No. But it will save the situation until further help can be provided. So that's what the initial response is, collagen type 3. Around the sort of 4th week, you have these fibroblasts and tenocytes, and they start producing type 1 collagen. And other cells start resorbing the type 3 because, they patched it up, okay? Now we need the pros to come in. We need people who can help even further. And collagen type 1 is what it's naturally there. Now, collagen type 1 is, they try to lay it in the line of the force. And what this means is how the force is applied to the tendon. So if the force is applied in this direction, you're not going to be creating, collagen in that direction. You're going to be creating in the direction of the force. Another way to think about this is you have your bone, right? That's your bone. And you have an attachment here. This is your tendon. And that's your muscle, and that's your tendon. Now, the tendon goes in this direction, yeah? So they're not going to be arranging the collagen in that direction, across, right? They're going to be arranging in the direction of the force of the muscle. And if collagen was arranged in the opposite direction, it would create a much weaker tendon. Now, so collagen type 1 is laid down in force. So we have all of this laid out, but it's chaotic, right? As we said in the bones, they lay it all down, but it needs to be resorbed. And the macrophages do this because you don't have osteoclasts and other cells which can, release matrix metalloproteases and other substances to break down the tendon. Now, the stability is acquired through physiological use of the tendon. So this is where physiotherapy comes in for the tendon. So it's physio. So when you move, so the tendon heals by when you move it, right? When you have an Achilles tendon, they tell you to go in tiptoes and go up and down with your tiptoes. That helps heal the tendon because it's being used. And if it's being used, it gets a little bit inflamed, but you also get blood flowing into it. Now, the key point here is the cross-linking between the fibrils. And this cross-linking here, it ensures that the collagen that was deposited, creates links, and it's like chains and armor. It's like chainmail, right? You have all these chains together which create a protection. Something similar here, but it's to do with the fact that the forces are equally applied. Now, full regeneration is never achieved because the collagen fibrils, the new ones, are thinner and the defect still remains. What does happen, though, is that the tendon is partially regenerated and you regain the access and the use of that tendon. Now, if you don't do your physiotherapy, you don't get stability, and your tendon does not heal. And that's when you have to move on to more drastic, more intense therapies. But this also highlights the problem of tendinopathy, because tendinopathy is a big problem for athletes, for overuse, and for everyday people. And because you can never regain the tendon you had before, it's quite a big problem. But understanding how the sort of tendon heals sort of helps us. Helps you answer questions, get the marks. But B also gives you a bigger appreciation of how much of a problem it is for these people. Their tendons, they try and they might do physio, but it never gets back to the original strength. So that's a sort of appreciation for the patient's injuries. Now that's all we have. That's all the questions. Oh, we have a question here. So does PDGF and TGF beta recruit the tenocytes? So if we go way back here, cells of the tendon. So TGF, what it does is that it helps differentiate the tenocyte, right? It helps differentiate the tenocyte to become a tenocyte from a tenoblast, and also helps drive the specification of the tendon cells. What PDGF does is that it attracts, it's chemotaxis, it attracts the right cells to get to the right place. So think about it. How do the cells know when, let's say, you get a cut to go to the right place? The way that works is that you release all these chemicals, and PDGF is one of them, which helps promote and recruit other cells, which then attract the tenocytes. Okay, someone wants me to go through the periosteum. Okay. So if you think about the periosteum here, I'll just delete all this. So the muscle itself, okay, periosteum linked to the tendon, fine. So the periosteum is like a wrap around the bone, right? And it's continuous with the peritenon or the paratenon, depends on the spelling, and the muscle. So the muscle itself is not exposed, right? We have this thing called fascia, which is also another word for the epimysium. Now, the muscle needs to be enclosed because if you get contractions and it rubs against the bone or against bursa or sometimes other muscles, you have other muscles together, think of the leg, think of the forearm, you need that to be able to work on its own. Now, it needs to be linked together somehow, right? And that's this epimysium. Now, as the epimysium goes towards the tendon, it becomes the peritenon. And then when it reaches the bone, right, it attaches to the bone directly, but you also have a covering around it all, which is the periosteum. And this is where you have your blood vessels. So you imagine your blood vessels here, you have blood vessels like this entering the muscle. Of course, the blood vessels for the muscles can also enter from other places. And so they go like this. They go through here. Because they can't go through a tendon. You know, I mean, you might have some through a tendon, but if you cut a tendon, right, it generally doesn't bleed because it's avascular. If you just cut it or cut it open, or with scissors or with scalpel, it doesn't bleed because there is no blood supply through there. So you need this peritendon, you need this periosteum for the cells and for the blood for all the cells around it. So you can imagine there's blood like this coming through, and also then venous blood up the way back. And the way it's attached is that it's continuous. So this is not what it looks like here in the diagram. This is not what real life is. You would have an extra layer like this. It's very thin. It's very thin. Think about it like a wrapper for the bone. It's very thin. But what it does is that it provides a continuous blood supply. It provides support to the underlying tissues. And like you can see this in if you buy chicken from a butcher, and you look at the bones and you look at the tendons, you will see that the bone is wrapped with something, and this is the sorrow. This is the periosteum, and the periosteum is continuous with this muscle fascia. Now, muscle fascia brings us a whole different thing, because you can sometimes get inflammation between here. So you might get some inflammation here. You can get inflammation there. And that's because you have the blood vessels there. And that's where massagers help, because you recreate the blood supply. But back to the link. The periosteum, it's connective tissue. Think about it as a wrapper, right? Think of it as in like it's like a plastic bag around it. But within the plastic bag, you have all these nutrients. You know, you have all these blood vessels which help regenerate the tissue. But the blood vessels, they do not go through the tendon, which is why the tendon is quite avascular, especially when you get older. The older you get, the more avascular, which means the less blood flows through the tendon. I hope that made it a bit more clearer. So yeah, so the periosteum and the perimysium and the peritenon is just wrapping. It's connective tissue that helps support the tendons. So with ligaments, it's a little bit different. So with ligaments, you get different. You're talking about this one here. So ligaments, you get different factors, but it's more or less the same. It's the same with the inflammation, same with the repair, same with the remodeling. It's just that the cells would be fibroblasts rather than tenocytes, because tenocytes are specific to the tendon. Ligaments, they would use slightly different factors, right? This would be slightly different. But the cells are more or less the same. Like, for example, in the cartilage, you have chondrocytes, right? Which are just fibroblasts that are in the cartilage, because chondro means cartilage. So as you can see, I mean, depends on who does the research here, but they're basically just fibroblasts. And fibroblasts are these general cell that builds. Now, if it's in the bone, it's called an osteoblast. If it's the tendon, it's a tenoblast. If it's which becomes a tenocyte. And if it's in the cartilage, it's a chondroblast, which becomes a chondrocyte. And these unique names, tenocytes, fibroblasts, and all these other things, they just exist to give specific terms to the cells. But these cells, if you look at them under the microscope, or even the antigens they present, they're very similar, right? They are very similar in their function. It's just that the local environment they're in is a little bit different, and it gives a little bit different chemicals. But it's the same process. It's still inflammation, repair, and remodeling. It would just be different cells doing it with a different name. But yeah, this slide roughly can apply to cartilage. It's still inflammation, repair, and remodeling. But with cartilage, it is it's type 2 collagen rather than type 1. And we'll be going over cartilage in a future video. Any other questions? If anyone else has any questions in the future, just write them under the video. and I'll answer them, or just write them in this document, and we'll get back to you. But yeah, basically, if you think about the whole thing, all the musculoskeletal tissues are very similar. You have soft tissues, which is your sort of like your ligaments and your tendons, and then you have your sort of harder tissues, which is your bones and cartilage. But the process is very similar, right? And if you think inflammation, repair, remodeling, IRR, that's sort of like a good way to think about the healing processes behind this. And A, it's good for memorization because it's quite simple. It's just 3 steps. And B, it's also, you understand the science behind how all these tissues heal. I remember that like the cytokines, the chemokines, all this stuff, interleukins, that it was quite tricky in first year. I did an intercalated degree in immunology, so I have a better understanding of them. But there's so many cytokines and chemokines, there's so many of them that learning all of them is, I would say, a waste of time, unless this is something you want to do in the future. What you need to know is that for certain scenarios and for certain healing processes, you'll have certain cytokines and chemokines. And learning them all is not necessary. For example, tendons, there's still research for tendinopathy. Is it IL-17? Is it IL-23? What's what? No one's really sure, but it seems to be IL-17, IL-23. But at the end of the day, that's not something you'll be examined on, and that's not something you really need to know. What is important is that you appreciate the fact that these things exist. And not mentioning PDGF and TGF-beta, okay, fine, you might not get full marks for it, but if you just say chemokines and cytokines, you're going to be fine. So yeah, that's all. We'll be giving, so this video will be replayable. We'll be giving you feedback. Oh, there's another question. Oh, no, never mind, no questions there. We'll be giving feedback to see what we can improve. Do let me know if you like the document, if that's good for questions, or if you would rather prefer, if you're okay with a live chat. That's up to you. We'll have links to the video description. And if you like this, like the video, if it was useful, if this is the sort of content you want us to create, continue making, then do subscribe. And we're doing more. There has been a form for what's going next week, and it'll likely be joints or embryology. So it's one of the two. So yeah, thank you everyone. I hope that was useful, and yeah, see you later.